Okay, students, let's walk through the Meselson and Stahl experiment. I apologize for my voice, I have a bit of a cold. So we've taken bacteria and we've grown them, the parent generation, in heavy nitrogen, and then we moved all the cells over into light nitrogen, which is that lighter blue color, and we let them replicate their DNA once and replicate their DNA twice. And these are the three hypotheses that scientists had. And so you can see if it's the parent cell is all grown in heavy nitrogen, so this would be what we would call a heavy strand. Does that make sense? This here would be a heavy strand. This here would be a heavy strand, right? But this strand up here, it's made of light nitrogen, so it's light, okay? And these are all light strands. Whereas if it's in the semi-conservative model, the first one was heavy, but these two, because they split in half of them is heavy and half of them is light. Does that make sense? And then when they split in half again, this one would be a heavy light, and then these would be lights, right? So if it's a heavy strand, if it's heavy, it's going to fall to the bottom of the tube right here, right? So in the parent generation for all three of these hypotheses, when you spin it down, it's so heavy that it falls to the bottom of the tube. Then if we look at this first replication for the conservative model, you can see that we still have that heavy strand, but then we've got this strand right here that's light, so it's going to end up at the top of the tube somewhere, right? And then in the third round, we have this heavy strand that's falling at the bottom, and then we have three of these light strands that are falling up at the top, right? In semi-conservative, we start with that heavy. In the second round, they're both a heavy light, which I'm just going to draw in blue for lack of another color, but they're somewhere in the middle of the tube because they're sort of half heavy, half light, right? And then we have two of those heavy light strands, and we have two that are light that fall at the top of the tube. And then in our dispersive model, this is the idea, I don't know who came up with this idea, that it just exploded into a million pieces, and then the pieces were put back together with pieces of heavy and pieces of light. So these two strands here that are heavy light would be somewhere in the middle, right? And then you'd have four of these heavy lights that would be somewhere in the middle, maybe a thicker band right here in the middle. So you can see that these three hypotheses are mutually exclusive. They look very different. So this was an ingenious way to figure out how DNA was actually copying itself. All right, so we'll look at the results here in just a second.